Hey guys, this is Kendall Terry, and this is the sensory system part 2, concerned mainly with, in this one, eye vision. How do we perceive what we see, which is evolutionarily one of the things that separates us from a lot of organisms that we find in nature. So the first is looking at the evolution of the eye. There's 4 phyla that have an evolved eye: the annelid, the mollusk, the arthropods, and the chordates. All of them actually have eye structures. So when we're looking at those, what we have found is that those eyes have evolved independently of each other. In other words, you've got 4 separate evolutionary events taking place at one time. This is called convergent evolution, where you have a structure that seems very similar. But when you look at how that structure evolved and the characteristics of that structure, you realize that those structures do not show a common ancestry. Rather, they show a common environment. So you've got organisms that are having to adapt to an environment where they need to see, or where seeing gives them an added advantage. And lo and behold, we get the evolution over time of what we consider to be sight. So what's the structure of the human eye then? That's the one that we primarily focus with. You can see here the human eye on the screen. Of course, you've got the cornea, that protective layer on the very front of the eyeball. This would be kind of where the contact lens sets, if you have contacts, that sort of thing. Under the cornea, you have the iris, and the iris is the colored part of the eye. The pupil, which is kind of like the hole, if you will, that light's going to travel through. The lens, which is on the other side of that pupil, which is going to help shape that light as it comes through into the eye. And then you've got the very back here in the back, the retina, this level back here. Retina is where we find the pigments that help us perceive light. And then, of course, we have the optic nerve and the blood vessels coming into the eye in the very back that helps us to send those signals back and forth to the central nervous system. So how does the retina then perceive light? Well, the retina perceives light with 2 pigments in the human eye: rods and cones. Rods are for black and white vision, and then cones are for high visual acuity and color vision. You can think of this as seeing in HD as your cones, whereas seeing maybe the way your grandparents or great-grandparents saw television as the rods, black and white. So here's the structures when you compare them. So when you're looking at the difference between rods and cones, you can see that. Of course, one, their shape obviously gives their name. Rods look like this, cones look like this. And then you have this outer segment where all the pigment discs are located. And then they're connected with a cilium right here to what we call the inner segment. In the inner segment, you have tons of mitochondria. You can see that in both pictures here. Tons of mitochondria, because those mitochondria are going to help send that signal. You need energy. And they're going to help send that signal with energy. Of course, the nucleus of that cell, and then finally the synaptic terminals, where they're going to send that through to a nerve cell to be sent on then to the brain. So how are signal transduction pathways used in light reception and transmission? In light reception, it's actually different than all the ones we've talked about so far. And all the ones we've talked about so far, there's been some kind of trip, and that trip then has caused an action potential to be reached and then sent. In vision, it's actually an inhibitory neurotransmitter that works the opposite way that we usually do. Let's see a picture here. This is in your book. But in this one, whenever we're in the dark, you can't see in the dark very well. So in the dark, your eyes will actually, cyclic GMP levels are high, and they keep chemically gated sodium channels open. So sodium influxes, this depolarizes the cell. This allows an influx of calcium ions, which leads to a release of inhibitory neurotransmitters, which, in other words, prevents the signaling of the bipolar cell. Here's the bipolar cell, and these are connected to those rods and cones. And this bipolar cell is being inhibited right now, meaning they're saying don't send any signal right now. It's in the dark. Whenever we're in light, however, the cyclic GMP is low because it's being converted to just GMP, and this causes then a rhodopsin here to activate a G protein that stimulates phosphodiesterase which converts that CMP to GMP, that cGMP to GMP, and in doing so, it closes, because then those gated ion channels are no longer getting the signal they need, it closes the sodium ion channels, which then, in essence, closes the calcium ion channels. It doesn't allow calcium to influx, so you get a hyperpolarized membrane. And this hyperpolarized membrane then prevents the release of inhibitory neurotransmitters, allowing the bipolar cells to then fire. So this is kind of opposite if you think about the way we've looked at other nerve cells. This is opposite the way they function. And whenever it's saying don't send it, it's actually, whenever they're saying there's nothing there to perceive, it's sending a inhibitory saying don't fire. And whenever there is something to see, it's not sending the inhibitory, so the bipolar does fire. So it's a little bit different way to look at it. So how are rods and cones then connected to receptors? In the fovea area of the eye, which if you go back to the picture that I showed you, you'll see a little notch in the retina called the fovea. And this is where the cones are located, and they make a one-to-one connection with a bipolar cell. And each bipolar cell synapses in turn with one ganglion cell, which is a direct site, which means that each cone sends a direct signal through the nerve cell coming into the eye to the brain. So you get a direct connection for the cones. Whereas in the rods, many rods will converge on a single bipolar cell, and many bipolar cells can converge on a single ganglion cell. So you don't have that. You have more of a periphery of vision. So if you think of that in the way you see, if you look straight on, you get a very good visual perception, and that's because of the fovea in the back of the eye, and it's perceiving and sending a one-to-one visual connection. However, you still have what we call a peripheral vision. You can still see off to the side, maybe not as clear, maybe not as acute, because the rods, primarily located on the sides and everywhere other than the fovea, you'll have they all are connected in kind of groups and clusters, so they're all sending these signals of understanding what they're perceiving out here in the periphery, but it's not as clear of an account as what the fovea is able to send. What about color blindness? And if someone says that they're colorblind, maybe they can't see red well, or they don't see blue well, or something like that. Well, normal vision is actually trichromate. And you can think of this kind of like your printer cartridge, maybe for your printer. It comes and it's got the three circles that kind of all interloop, and from those three circles of colors, you get all the different colors that your printer is able to print. Same with your eye. You've got three versions of cones in your eye that perceive the three primary colors, and then those three primary colors in whatever version that's being tripped will help you get all the different colors that you see. So you are called a trichromat if you have all three working cones. If you don't, and maybe you have one cone that isn't working right, then you would be a dichromat, meaning you're only getting really two colors that you see well, and then everything else is a variation of those two colors. So you have something maybe that you don't perceive very well. Maybe it's a very dull color, or it's like a gray or whatever. It's not a color at all in what we would consider to be an actual color. So there's an issue there. That's color blindness. We'll talk more about that possibly later on.